I want to thank Dr. Ratner, Dr. Himmelfarb for the invitation. Truth is sort of said, I'm a vascular surgeon, so it's clearly a different meeting for me. But to be honest, it's been horribly enjoyable. As a sort of a senior vascular surgeon, I've been to my share of clinical vascular surgery meetings, and I sort of welcome going to meetings where I get sort of a different perspective and sort of different ideas. And one of the things I've really taken away from this meeting already is sort of the way the meeting is really integrated, sort of patient engagement, patient perspectives, in a way that I think, it's underserved in the vascular surgery community. I'm sort of scratching my head thinking about sort of how to sort of create this kind of environment in some of our meetings. So really a wonderful meeting, and sort of more to come today, I'm sure. The other thing I'll say is when I was sort of emailing Jonathan and saying, what would you like me to talk about? I could talk about, a vascular surgeon's perspective from access. And he said, well, I can also talk about some scientific things. And he said, well, given the sort of the nature of the first two talks, why don't you talk about some scientific things? So I said, a couple of different topics I could talk about. And he said, why don't you talk about, both topics? And in this case, hemodynamics and cellular adaptations. So to be honest with you, over the last 3 days, I've sort of scratched my head trying to figure out how to put really two divergent topics together into a single talk in a way that I hadn't done before. So hopefully it sort of makes some sense. Oh, a clicker. Sorry about that. Is that the right one? Oh, perfect. So this is just sort of an article we published recently in JAMA Surgery, taken from the HFM data, which sort of underscores sort of the initial primary patency with fistulas is in the 45 to 50% range, whether you're already on dialysis or, dialysis is starting for you sometime in the future. And we can salvage another 20 or 25% of these with some type of intervention, which sort of leaves, obviously, a lot of these that fail to go on to mature. So, my background is in engineering, actually, so I think of sort of driving forces and things like that, and really ask the question, what's the primary driver for fistula maturation? And when you think about sort of engineering things, you think about sort of this kind of homeostasis between an environment and biologic systems, and one of the primary drivers in fistulas is shear stress. So if you take a vein and you sort of expose it to either low or high shear stress, you get sort of an inward constrictor remodeling or an outward expansive remodeling. And it's sort of this balance between inward-outward remodeling and intimal hyperplasia that really dictates the outcome, and Prabir touched on this to some degree. So high shear stress causes enhanced outward remodeling, also sort of leads to minimal or decreased intimal hyperplasia. Conversely, low shear stress does the opposite. So it's really a self-regulating system if everything works correctly. We wanted to test this in patients. So as an offshoot of the HFM study, for those who don't know, the HFM was the Hemodialysis Fistula Maturation Consortium. Dr. Himmelfarb was involved in the University of Washington, but 6 centers across the country sort of enrolled about 600 patients within that study. But an offshoot of it was a 120-patient study where we did serial MRI scans. So we did MRI scans at 1 day, 6 weeks, and 6 months, and from that we were able to extract both the geometries as well as the flow dynamics, and we could do relatively sophisticated computational modeling. Within this, we took all fistula types that were one stage, and about a third were in the foramen, and 2/3 were in the upper arm. This just shows the pipeline of how we go from the MRI images and the data we collect to really get the full map of the hemodynamics. So this is one of the primary results of this study. In fact, this manuscript is sort of under secondary revision now, and hopefully within the next week or two will sort of be accepted for publication. But this is showing the vein, and it shows kind of what you'd expect or what we hope to see. Hang on. Is there a pointer on this? Is that a pointer? Yeah, there we go. So what we see is if one is sort of no inward-outward remodeling, and on this is shows the shear stress. What we see in this early phase is that the higher your shear stress, the more outward remodeling you get, or the more lumen preservation is what we actually measured. In the later phase, week 6 to 6 months, we also get sort of what we expect to be an effect where we get outward remodeling with increasing shears. You can see that most of the remodeling occurs in this early phase, but sort of continues on in this later phase as well. And we also had the opportunity to look at the artery, because I think, when you think about fistulas, it's not just the vein, but it's the artery sort of has to dilate and sort of be able to carry the flows, and we see a similar kind of pattern with the arteries. So I think it really asks the question, if we have an AV fistula that has normal physiologic remodeling, why do so many of these fail to mature? And it has to do with the fact that the flow dynamics are really off the chart severe. And what do I mean by that? So here is two computational fluid dynamic simulations. The top one is a superficial femoral artery. The bottom one is a brachiocephalic fistula. And what you see is a lot of sort of lighter and dark blue up here, which is relatively low velocities across the superficial femoral artery. What you see in the brachiocephalic fistula is you see a huge amount of sort of red and yellow, which is the higher velocity. So actually, the amount of physical force you see here is really huge. There's almost nowhere in the vascular system in which blood vessels are exposed to the kind of forces they're exposed to in brachiocephalic fistulas. This is sort of a picture which gives you a sense of sort of the temporal dynamics of this, and you can see how complicated this sort of flow system is. So actually, if you calculate the shear stress, it's about 10-fold higher in the arteries than you would normally see in the fistula vein than you would normally see in the arterial system. There's a whole lot of sort of flow, recirculation, stagnation because of the sort of the marked changes in geometry. There's vortex formation. There's a sort of transition to turbulence. Turbulence is really sort of a bad thing. It's something that has a lot of energy implicit in it, and that energy is dissipated into the wall, causing sort of a chronic injury. And the force vectors are really non-axial, meaning they sort of point into the wall as you come around these bends in a way that it causes further damage. If we do, the movie isn't playing, but if we, this is a slow motion movie that would show that these little sort of pockets of wall shear stress really sort of spin off, and they're very high frequency sort of spin-offs that occur. This is a fast Fourier transform. It's sort of an engineering way to sort of look at the frequencies at which sort of the changes are going to occur. And if you sort of looked at a normal artery, you would see everything in sort of this low frequency range. But when we look at fistulas, we see these huge high frequency pieces. So it's this high frequency pieces that sort of spin off, causes really a very chaotic, random kind of damage. So that's what really, I think, leads to a lot of the heterogeneity we see within these fistulas. So what are the implications of these severe and chaotic flow patterns? I think if we go back and we sort of look at fistula lesion location, and we've heard this in both our previous two talks, and that it's this swing segment, sort of the juxta-aortic segment, that is really important. About 63% of this 50% is sort of this swing segment where we connect the artery to the vein as being the area that leads to significant stenosis. So what we did within the study is we said, okay, what geometries were potentially good and bad? So we looked at anastomotic angle. We looked at non-planarity, which has to do with how much things diverge or curve. And we also looked at, sorry about that. Go back. We also looked at the anastomotic plane. So we looked at 3 variables. Interestingly, when we looked at the effect of these geometries on maturation, it really didn't have much of an effect, and you can see the p-values were all non-significant. But when we looked at the effect of these geometries on restenosis and the need for reintervention, we see that there was a lot of significance in terms of both an anastomotic angle and the non-polarity angle, sort of underlying the idea that it's the secondary stenosis at these swing regions that are really most impacted by the hemodynamics. So just to sort of talk about, summarize what I've said today, is despite the marked elevation in wall shear stress, the AV fistulas seem to have a normal adaptive response with high shear leading to outward dilation and sort of preservation of the lumen. The hemodynamics of this juxta-anastomotic swing zone are complex and chaotic. Anastomotic geometry and its impact on the hemodynamics is probably more important in stenosis development. And I think the optimum fistula geometry from a surgeon's perspective is to reduce the curvature of the proximal vein and maximize overlap. And some of the things I've taken from this is normally when you create a surgical anastomosis, bringing it just into the side is probably not the optimum configuration. Sort of reducing the sort of the angle of this curvature so that they're more collinear and doing really two things: extending your incision just a hair distally so you can shift this anastomosis a little more distally, and doing a little more vein mobilization so this vein can make a little more gentle angle, I think makes sense. So now what's the biology through which shear stress regulates vascular adaptation in a fistula? So when we think about shear stress, which sort of occurs at the inside of the blood vessel, which is lined by endothelium, that sort of does a whole bunch of things. It activates things called transcription factors within the endothelium. Also, those activation lead to growth factor elaboration and cytokine elaboration by the endothelium. It leads to the smooth muscle cells, or in the sort of deeper in the wall, and this is what provides integrity to the blood vessel. So there's different phenotypes. There's a contractile phenotype and sort of a more of a wild card, one that's sort of not really contractile but has sort of altered physiology. We call it dedifferentiated smooth muscle cells, and I'll talk about this in a minute. And then we have inflammatory cells, various leukocytes that come in, and it's sort of the interplay of all these pieces that I think is critical to understand. So one of the things I've become less enamored with sort of over my career was animal models and fistula work. I think, both the hemodynamics and the biology of the animal models are really poor surrogates for what goes on in sort of the actual fistula that goes on within patients. So as a surgeon, I think one of the things that I potentially have access to is how can we sort of use things we do in the operating room to sort of provide further understanding. A two-stage basilic transposition fistula is sort of relatively standard. This can be done either as one stage or two stage, and I think the movement in the most surgical practices has been to do it as a two-stage operation. So this is where in the first stage we create a simple fistula, in the second stage we transpose that fistula. And the way I do it is I sort of disconnect this anastomosis to sort of get rid of the swing zone, because I think this is problematic, and I reconnect the arterialized or the matured vein a little higher up on the artery. So it gives us the unique opportunity to collect specimens at sort of 2 points in time. We can collect it both when we place the initial fistula and then 6 weeks later, which is when we normally do the second stage. About 20% of the fistulas in our practice are created this way. So what it allows us to do is something called single-cell RNA sequencing. So every cell has this stuff RNA, which then basically controls the programming of the cell. One of the sort of the really new technologies is trying to look at cells on an individual basis and understand how this goes on. So 10X is the system that we use, and this is just sort of the roadmap for how this is technically done. So the first question is, I think, what happens to the artery? So we have an artery that's just exposed to normal flow, and then we sort of create a fistula, and it becomes in a significantly elevated flow. So what happens there? So first thing that happens is the baseline flow is in the range of about 70 mLs per minute. It goes up to over a liter a minute. So this same blood vessel now had a 15-fold increase in flow rate, and that's about a 10-fold increase in shear stress that occurs. So these huge changes that occur essentially immediately. This is a map. So each of these dots is a cell. And so this is what you get, is you basically know the full RNA composition within each of those dots. Essentially, with each of those cells, there might be, 20,000 different RNA sort of types within each of these dots. So it's a huge amount of data, terabytes, so really vast amount of data. So the first sort of step in this is you just basically know all these cells, and this is sort of a decomposition methodology that's done mathematically to try and sort of look at differences among these dots. And the first step is really to identify which dots are which. So there are sort of known markers for different cells, and then we could say, okay, let's sort of look and see which markers are in different cells, and we can basically look at these. These are the maps I just showed, the dot maps, and this we can use this to identify the different cell types. So we could see here, these are the smooth muscle cells, endothelial cells, macrophages, a small amount of uncharacterized cells, and fibroblasts is sort of being the dominant cell types. I think one of the interesting things that we can ask is, what are the different cell compositions to occur? First, we see, I expected actually some kind of sort of divergence of endothelial cells and see really sort of two populations emerge, but we don't. We really just see one population that stays with about the same number of cells. But I think when we look at the smooth muscle cells, we see a clear sort of shift from a contractile smooth muscle cell phenotype to this modified smooth muscle cell phenotype. So this sort of altered smooth muscle cell, which is very active, secretes a lot of different factors, and at least from a biology standpoint, is thought to be really deleterious potentially. The other thing we could sort of talk about is where are the changes in gene expression occurring. And surprisingly, again, we didn't see a lot of changes in the endothelium. The endothelium really didn't, not to say that the changes in response to the shear aren't important, but they're species-wise, there's not a lot of them, where we can look and see with the smooth muscle cells, huge sort of modifications that are occurring. And then we could take the endothelial cells and we could sort of do something called a gene ontology analysis, which looks at the different processes that are occurring. And we could do something called a KEGG analysis, which looks at the different pathways that might be changing between the first and second stage. And the blue is downregulated. So interestingly, what we found is that in the second stage, there's a lot of downregulation of pathways in the endothelium as opposed to a lot of excitement or activation which goes on, which I think is surprising and not what we entirely expected. The other thing I touched on briefly was that we're characterizing the smooth muscle cells. So I would say, actually the pioneering work was done here at the University of Washington. Cy Schwartz is one of the really people in vascular biology, and Mark Reedy and the group here really pioneered this concept of smooth muscle cell phenotype. So there's a contractile phenotype and a synthetic phenotype, and this has been sort of postulated in cell culture and some animal models for probably 40 years, I would say. In the last 10 years, maybe even a little less, there's been sort of further understanding as we've the molecular biology techniques have sort of advanced, and we really find that it's a continuum of different ones. So you have a contractile phenotype, and you have sort of various modifications towards a fibroblast type lineage, and you even have some that sort of create bone or some that are more like adipocytes. And we've really defined the markers which seem like define each of these. So we can ask that with sort of our group. And what we find is that we clearly have a contractile group of smooth muscle cells and ones that are sort of somewhere between the mesenchymal and the fibroblast phenotype. So now we sort of can think about, okay, these are the kind of phenotype changes that are occurring as we think about sort of beginning to modulate the biology. I won't. We can. You could go on and talk about the biology for ages, and I'll sort of spare everybody that. But we can look at the differences between contractile smooth muscle cells, fibroblasts, and modified smooth muscle cells to really understand. What are the ontology differences and what are the pathway differences? So again, we can sort of begin to think about how the response occurs in patients. Transcription factors are the things that sort of drive a lot of the biology. They're things inside cells that seem to activate a lot of the way the cell reacts. So we can actually go through and we can say, okay, where there are transcription factor changes between the two stages, and we see again that there's a lot of sort of contractile smooth muscle cell transcriptional changes occurring and very sort of quiescent in terms of the endothelial cells and the other cells. We can also begin to break down cell-cell communication, and we can say, okay, what are the differential interactions that are occurring? And we see that the communication between the smooth muscle cells, both synthetic and contractile, is really sort of upregulated. So these two cell types are talking to each other. And also they're talking to the fibroblasts. And there's some sort of backfeed between the endothelial cells and the fibroblasts between the first and second stage that sort of differentiates the way these cells communicate with each other. And then we can even sort of go on and say, okay, are they, is it direct contact, or is it some type of matrix-mediated contact? And we can begin to ask these kind of questions. So moving on to the vein, and how does the vein change in response to shear? First, if I was to show you histologic pictures of the artery, it would just look like normal artery. You couldn't really tell the difference histologically between the first and second stage. This is the vein. So these are the first stage veins that were collected, and these are the second stage veins. And you could see that there's a huge amount of heterogeneity, both in terms of sort of the initial vein as it looks like before it's used to create the fistula, and then what it looks like later on down the line. So this is the map I showed. That's where we went on and we identified the cell types. And interestingly, we found really three kind of varieties of endothelial cells that were present in the vein, even at baseline. So mostly was this one endothelial cell type, but two other cell types. And as we look at the smooth muscle cell fibroblast cell types, what we see is sort of a reduction in the amount of fibroblasts, but we don't see the huge sort of shifts in the smooth muscle cells that we saw in the artery. So really the vein is sort of adapting in a different way. We also saw these proliferating cells, which we really couldn't identify what they were, except that they really looked like they had a lot of markers for proliferation. And one of the big things is we see this huge influx of macrophages in the artery in response to the change in flow. Really no leukocytes to speak of. Macrophages, up to over 12% of the cell population is now macrophages. So again, sort of really a dramatic difference. I think is the first time this has been shown in the human setting. We can also do something called an unsupervised cluster analysis, where we look at sort of the top genes, and it's interesting to see sort of what clusters together. And we see these proliferating cells are probably some type of macrophage. We see that these endothelial cells kind of group together, and we could see sort of the smooth muscle cells. So this gives us an idea of sort of what the different cell types are like, at least biologically. If we look at the number of genes changing between the two stages, you know, these are huge numbers. And you look at the endothelial cell gene changes where they were basically teens, and I'll show you that in a second. So these are the artery gene changes, which are 36. And here we have over 1,000 endothelial cell changes. So the endothelium in the vein is hugely different than it is within the artery, and it probably has to go back to sort of the hemodynamic pictures I showed before, and those high forces that these arterial endothelial cells are shown to. And I think this understanding the biology of these endothelial cells and sort of being able to manipulate it for therapeutic use, I think is where we're going. And I think Prabir a little bit touched on this in his talk, where he was talking about how sort of understanding each patient and how you might sort of use this information to design specific therapies. Transcription factor analysis, kind of the same thing. Really a lot of changes in the endothelial cells that we never saw in the artery. So this kind of comparison, I think, really provides insight into these issues. So I won't go a lot into the biology, but just to say the dynamics in the vein are much more so in the artery, especially as you look at the endothelium and the macrophage leukocyte populations. Arterial remodeling is dominated by changes in smooth muscle cells, less so in the vein. Arteries seem to respond by changing the composition. Vein seems to respond more by changing their cellular phenotype. So a difference there. And interestingly, the pathways seem to be upregulated in the vein where they're downregulated in the artery. So this is the vein, and the red is upregulated, and the blue is downregulated. So it seems like the artery is able to dampen its response and sort of keep a normal structure. The vein basically goes crazy and with a lot of upregulation in a way that you get this really aggressive type of modification. So where do we go from here? So what are my next steps? So I think we need to define the cell-specific changes that determine adaptive versus maladaptive remodeling, i.e., fistula success or failure. And what do I mean by that? I mostly talked about sort of artery and vein and how they're changing, but what we really want to know is if we have a fistula that works, what's its cell phenotypes? If we have one that doesn't work, what are its cell phenotypes, so we can begin to sort of engage it at that level. And I think we need about 50 to 75 patient samples to make a sort of a reasonable sort of shot at that. We're about halfway in terms of getting there, so the recruitment continues in that regard. One of the real critical things that is going on in this study that I didn't sort of describe is we do high-resolution ultrasound, and we do sort of histomorphology on some of the tissues we collect, and we do some back table sort of reconstructions of these. So we're able to develop the in vivo compartmental sort of volumes in areas in a way that we know, okay, was this expansive remodeling? Was this hyperplasia? And how do these things sort of play into each other? So it's this kind of understanding. We're not going to be able to just talk about did a fistula work or not from a dialysis point of view, but we can sort of focus in on the biology and understand what happens in terms of sort of the morphology of those fistulas. So the last thing is understanding compartmental differences is going to be really important here. And by that I mean, what's going on in the smooth muscle or smooth muscle smooth muscle-like cells in the intima versus media. And what do I mean by that? I mean, so here's a fistula that demonstrates expansive remodeling. This is one that sort of demonstrates aggressive intimal hyperplasia. If you ask biologically what we've asked this vein to do to create a useful fistula, well, we've asked it to break down its extracellular matrix and reform it. We've asked it to increase its number of smooth muscle cells, and we've asked it to reorganize its smooth muscle cells to maintain a viable structure. The ones that sort of we asked that demonstrated aggressive intimal hyperplasia, kind of the same things are going on. There's a lot of matrix deposition. There's a lot of increase in the number of smooth muscle cells, and these smooth muscle cells are migrating from the intima, or from the media and the intima, causing this hyperplastic lesion. So it's interesting that the biology almost overlaps each other. So there's subtle differences, and it's sort of this balance that occurs between these various processes and understanding the biology in depth, because just on the surface, it's the same things that are going on, but they're going on, they're having sort of less profound differences between these two that are making the effect different. And what we're, so there's the next generation of this, and something we're going to engage in over the coming months, is something called spatial transcriptomics. So this is basically, I don't know how well it projects, but each of these areas is a little dot. And this is a histologic section, and what each of these dots is 10 microns in size. So essentially what it does is it allows us to not only know what the cells are, but it allows us to know where they are and how they're sort of oriented within a tissue. So it's essentially each of these dots will provide a full transcriptome, similar to each of the individual cells do, in a way that we can begin to understand, okay, in the media, those smooth muscle cells have this kind of sort of changes that are maladaptive or bad, and in the intima, they might be vice versa. So that kind of detailed spatial information is really, I think, the next phase of this. So I neglected to put a acknowledgement slide in as I was sort of just figuring out what to talk about, but obviously a huge lab effort. Most of this work has been funded graciously by the NIH, NIDDK. And people in my lab, Yong He and Jared Rosowski, are sort of the people who've done the hemodynamics as well as the single cell work I described. And then sort of partners. None of this is done without sort of external partners. Prabir, I don't know if he's still on there, but Prabir has sort of been a participant in all the hemodynamic work we've done over the years. And then in Utah, Alfred Chung and Yan-Ting Xu are sort of the group there that have really fueled a lot of this. So with that, I appreciate the invitation.
So, yeah, thanks, Scott. That was really very informative. We're a little behind. We had a group Q&A session, but I had questions after each talk, so I don't think we need the group session. In any event, if there are any questions for Scott, we certainly can one or two questions. Yeah, please.
Yeah, I have a question. One of the reasons that the blood flow rate in AV fistula is high is because the dialysis works much shorter than the native kidney. And I know there's a practice that if the patient receives a kidney transplant, then the doctors will try to clamp down the fistula to slow down the flow rate. I'm wondering, in the future, suppose we have a 12-hour per day dialysis mechanism, is there a benefit to use smaller diameter fistula?
Yeah, it's an interesting question. When I know, and other surgeons and sort of the studies would say that if you start out with a vein that's smaller, its chance of sort of becoming a mature fistula is less. But on the other hand, as you use bigger veins, then the resistance is less and the flow rates are greater. And I think those are sort of intersecting pieces that are really at odds to each other. So I think there is sort of some sweet spots and some reconstructions that from a surgical perspective or even an endovascular perspective that offer those opportunities. And I think one of the exciting things, I think, from a surgeon's perspective about the endovascular technologies is that it probably is a way to create a fistula that's got lower flows, but apparently sort of the ability to have the same level of maturation and patency. Now, we know there's no comparing apples and oranges, and that the veins that were used in a lot of those studies were really sort of optimum veins. But nonetheless, I think it's the concept that there is probably a sweet spot in terms of creating a certain amount of flow, but still having a vein that's big enough to sort of provide a useful fistula. And I think one of the things from a surgical perspective, for example, and I think sort of many surgeons will do this, if you're creating a fistula in the antecubital fossa, sort of taking that vein and sort of mobilizing it onto the radial artery. So the artery itself sort of then creates a little bit of a governor to it. So I think the thought of sort of banding a fistula up front in its initial creation sort of scares a surgeon too much to do it, and that you just want to get a fistula that works, and sort of to monkey around with creating some banding or something, just makes us too nervous.
We have a question on Zoom. To what extent are these cellular changes in the AVF caused by changes to tissue stiffness as compared to shear stress?
Yeah, I think you sort of talk about what you can talk about and you avoid what you can't talk about. And that's I think that's a really important question, and that the stiffness and sort of the compliance mismatch in all of this is really important. And I talked about sort of this vibration that occurs in these high-frequency oscillations. I think really that causes not just shear stress, but it causes a lot of sort of mechanical motion, and the stiffness aspect is really important there. The problem with sort of modeling those kind of things is we can non-invasively get flows pretty well, and we can get geometries, and that's how we can understand shear stress. But to understand sort of this interaction between hemodynamics, blood flow, and the wall structure biomechanic pieces, you need to have a lot more information. You need to have an understanding of the mechanical properties of those veins in a way that, and you have to sort of understand or have sort of imaging modalities that provide sort of high resolution movement, all of things which are very difficult to do in patients or humans. So I think a really important question, just a really hard one that we don't have the technologies to engage with at this point.
Scott, could I ask quick questions? First of all, that was a great talk. I think you've come closer to the truth in vascular access than anyone else. I agree completely that the truth lies in human studies. But a question that I have is how much of all the changes and amazing changes in the vein in particular are linked to uremia as opposed to just hemodynamics? Because perhaps dissecting that out could point us in certain directions for therapy as opposed to others.
Yeah, fair question. As you know, I mean, trying to sort of understand that at the patient level, because patients are getting fistulas if they don't have sort of the uremic aspect of it, and sort of I think the shades of gray of uremia that occur in patients who are getting fistulas probably not a way to answer it. Animal models, as your lab has done, is sort of the way to sort of get out of that, sort of address that. So I do think. Trying to sort of understand what's going on sort of in patient-level studies and then bringing that back into the animal. I poo-pooed animal models a little bit, but I do think sort of understanding patients and then understanding the animal models from sort of a detailed biologic perspective at this single-cell level, I think is going to really enrich our animal model work going forward.
The truth is always in humans, Scott. You are correct.
But it's you do what you can do, and sometimes you can't do it, and you got to recognize that.
So, Scott, first of all, I want to really congratulate you for bringing an incredible level of science to the simple act of connecting a artery to a vein and the complexities that are there. We really do need this level of science if we're going to make better vascular access. The question I have is, before there was the hemodialysis fistula maturation study by the NIH, as you know, there was the Dialysis Access Consortium study, and the goal of that study was to come up with pharmacologic agents that could enhance AV maturation, and those studies failed. Armed with the new knowledge you're creating, are you getting any clues for pharmacologic agents that could enhance AV fistula maturation in your estimation?
I would say, that's obviously the ultimate goal for it. At this point, nothing that I think I would sort of engage with at this point. I think, getting the full data set of 50 to 75 patients and sort of asking that question is what differentiates success and failure is really sort of, I think, going to point us in those directions. I would say that of the things that I think are really important, I think, inflammation and macrophages seem to be a real driver of it, and we talked about that when we had a meeting on Sunday evening and talked about the importance of inflammation and all this, and I think that really underscores this, is I think, you could think of sort of dampening inflammation in sort of some kind of global way with, some sort of, corticosteroid or other, some kind of anti-inflammatory, maybe aspirin or something has, in addition to his antithrombotics. But really targeted sort of modulation of that inflammation is, I think, number one. And then two, trying to engage sort of these sort of early endothelial cell activation that might occur in veins. And how do we begin to sort of dampen that sort of shift or sort of that really aggressive activation of the endothelial cells, I think, is where the main payoff is going to be.
Thanks.
Thanks.